400 million years ago, every continent on Earth was a desert. Not because of drought, because nothing had figured out how to live there yet. Then, in a geological instant, something changed. Plants left the water, and in doing so, they did not just colonize the land. They rebuilt the atmosphere, invented soil, triggered a mass extinction, and accidentally created every forest that has ever existed since. Before we get down to business, please subscribe to this channel. Your support really helps us to continue this work. Thank you. To understand what the first trees were, you first need to understand what the world looked like before they existed. And that requires a genuine act of imagination, because the world before land plants is so different from anything in modern human experience that no landscape photograph, no nature documentary, no painting can quite prepare you for it. Picture the most barren, lifeless stretch of rock you have ever seen. A high alpine scree field, perhaps, or a fresh lava flow in Hawaii, or the surface of a dry riverbed in the middle of a continental desert. Now remove the occasional hardy plant clinging to a crack in the rock. Remove the lichen on the stone surfaces. Remove the invisible bacterial films that coat every rock surface in the modern world. Remove the soil entirely, because there is no soil, just mineral particles. And then extend that barren, chemically weathered rock surface across every continent on Earth, from pole to pole, coast to coast, for hundreds of millions of square kilometers, broken only by rivers and lakes and the margins of the sea. That was the Ordovician and early Silurian world. That was Earth before plants. The oceans were alive. The Ordovician and Silurian seas were extraordinarily diverse, filled with trilobites and cephalopods and echinoderms and the first vertebrates, a complex marine biosphere that had been building for hundreds of millions of years. But the land was empty, not merely sparse. Empty. There was weather, certainly. Wind, rain, rivers carving channels through bare rock, glaciers advancing and retreating. There was a water cycle. There was a rock cycle. There was geology happening in the normal way. But there was no biology on the land surface of any significance. No animal had any reason to leave the sea, because there was nothing on land to eat. No shelter, no shade, no accumulated organic material of any kind. The land was a sterile mineral substrate waiting for something to colonize it. And for the first 4 billion years of Earth's history, nothing had managed to do so in any meaningful way. The first organisms to make a serious attempt at land colonization were not plants in any sense we would recognize today. They were, in all probability, a partnership. Symbiotic associations between fungi and photosynthetic organisms, either cyanobacteria or early algae, forming structures that are the ancestors of modern lichens. These ancient biological crusts, thin films of living material coating rock surfaces in areas where moisture was periodically available, were the pioneering organisms of the terrestrial world. They could survive desiccation. They could tolerate temperature extremes far beyond what any aquatic organism could manage. They produced acids that very slowly dissolved the rock beneath them, releasing mineral nutrients, beginning the geological process that would eventually, over hundreds of millions of years, produce soil. They were humble, nearly invisible, operating at the boundary between geology and biology. And they changed the world as surely as any more dramatic evolutionary innovation. Alongside these early biological crusts, the ancestors of mosses and liverworts were making their own tentative experiments with terrestrial life. These early land plants, or embryophytes as botanists call them, shared a fundamental challenge with every organism attempting to leave the water: the problem of desiccation. In an aquatic environment, an organism is surrounded by the substance it needs most. Water is everywhere. Nutrients diffuse in. Waste diffuses out. Reproductive cells can simply be released into the water column and carried by currents. On land, none of this works. Water must be conserved. Nutrients must be actively acquired from a solid substrate. Reproductive cells released into the air will dry out and die unless they have some protection against desiccation. The entire body plan of aquatic organisms, optimized over hundreds of millions of years for a water-saturated environment, is simply wrong for land. The solution that early land plants evolved was a suite of innovations so fundamental that they remain the defining characteristics of all land plants today. A waxy cuticle covering the outer surface of the plant body, reducing water loss. Stomata, tiny pores in the cuticle that can open and close to allow gas exchange while minimizing water vapor loss. Spores with tough, chemically resistant walls that can survive desiccation and UV radiation during dispersal. And in the most critical innovation of all, a life cycle that alternates between two generations, one dependent on water for reproduction and one that is not, allowing the plant to exploit terrestrial habitats while retaining the aquatic reproductive mechanism as a fallback. These were not simple evolutionary changes. They were a complete reimagining of plant biology achieved over tens of millions of years through the usual process of random mutation and natural selection, but directed by the relentless pressure of a new and extraordinarily challenging environment. The earliest fossil evidence of land plants dates to approximately 470 million years ago in the Ordovician period, in the form of fossilized spores with the chemical and morphological signatures of land plant origin. These early plants left almost no macrofossil record, partly because they were small, partly because terrestrial environments are poorly suited to fossil preservation compared to marine environments, and partly because the organisms themselves were so simple that their physical remains were easily destroyed by weathering. But the spores survived, and their survival in the geological record tells us that something was living on land, something plant-like, something producing resistant spores in a world that most people imagine as entirely bare. By the Silurian period, roughly 440 to 419 million years ago, land plants had made their first major evolutionary leap. Fossils from this period preserve the remains of plants with a genuine vascular system, internal tubes for conducting water and nutrients through the plant body, a feature called the xylem that is so fundamental to modern plant biology that the organisms possessing it are called vascular plants, or tracheophytes, and they include virtually every plant species you have ever seen. The invention of the vascular system was transformative because it solved the single biggest limiting factor on plant size in a terrestrial environment: the inability to move water from roots to leaves against the force of gravity. In a moss or liverwort, water moves through the plant body by diffusion and capillary action, processes that work over distances of a few millimeters to a few centimeters, but fail completely at larger scales. With a xylem, water can be pulled upward through the plant body by the evaporative tension at the leaf surface a mechanism called transpiration pull that works against gravity over distances of tens and eventually hundreds of meters. The vascular system made height possible. And height, in the relentlessly competitive world of photosynthesis, is everything. The earliest vascular plants in the fossil record are organisms like Cooksonia, a plant so simple that it challenges our intuition about what a plant should look like. Cooksonia had no leaves, no roots in any proper sense, just simple anchoring structures called rhizoids. No flowers, obviously, just a branching stem, Y-shaped in its simplest form, with spore-bearing capsules at the tips. The entire above-ground structure was green and photosynthetic because there were no leaves yet to take over that function from the stem. The whole plant was perhaps a few centimeters tall, barely ankle height, growing in dense mats in wet habitats near water. But it had vascular tissue, it had stomata, it had a cuticle, it had resistant spores, and it was, in its small, simple, easily overlooked way, the beginning of something extraordinary. If you walked across a Silurian landscape, and if we imagine a landscape hospitable enough to walk across, which was only possible near water, you would see these small vascular plants as a pale green fringe at the water's edge, a centimeter or two of vegetation giving way immediately to bare rock and mineral substrate. The landscape would have been overwhelmingly dominated by rock and sky and water. No shade anywhere, no undergrowth, no sound of birds or insects, because nothing that could live on land had yet evolved to do so. Just the wind and the water and that thin green fringe hinting at what was coming. And then the Devonian arrived, and everything changed. The Devonian period, running from approximately 419 to 359 million years ago, is sometimes called the Age of Fishes because of the extraordinary diversification of fish that occurred in its seas. But from the perspective of land biology, it should be called something else entirely: the Age of Invasion. Because in the Devonian, land plants did not simply persist at the water's margin. They exploded. They diversified. They grew taller and more complex. They developed roots and leaves and increasingly sophisticated vascular systems. They moved away from the water's edge and began colonizing drier ground. And by the end of the Devonian, they had achieved something that had never existed before in the 4 billion year history of the Earth: a forest. The pace of this transformation was breathtaking by evolutionary standards. In the space of roughly 60 million years, land plants went from centimeter-scale leafless stems at the water's edge to 30-meter trees covering significant areas of multiple continents. That is a factor of 3,000 in height, achieved in 60 million years through the gradual accumulation of anatomical innovations that were each individually small but collectively transformative. To understand how this happened, you need to understand the specific engineering problems that had to be solved before a plant could become a tree. Because a tree is not simply a large plant. A tree is a fundamentally different kind of organism, subject to physical constraints that don't apply at smaller scales and requiring specific biological solutions to each of those constraints. The first and most fundamental problem is mechanical support. A small plant near the ground is supported partly by the turgor pressure of its own cells, essentially the pressure of water inside each cell pressing outward against the cell wall, and partly by the structural properties of its vascular tissue. This works fine up to a certain height, perhaps half a meter or so. Beyond that, gravity becomes an increasingly serious problem. A taller plant bends and breaks unless it has structural materials stiff enough to resist bending and strong enough to bear the compressive load of its own weight. In engineering terms, you need a material with a high Young's modulus and adequate compressive strength. Modern trees solve this problem with wood, a composite material made primarily of cellulose fibers embedded in a matrix of lignin, which gives wood both its stiffness and its remarkable resistance to compression. But wood did not exist at the beginning of the Devonian. It had to be invented step by step through the evolution of increasingly lignified cell walls in the vascular tissue of early land plants. Lignin is one of the most important biological molecules in the history of life on Earth, and it is worth spending a moment on it because its significance goes far beyond structural support. Lignin is a complex polymer, a large molecule made of smaller subunits linked together in a three-dimensional network, and it is extraordinarily resistant to degradation. It is not easily broken down by most bacteria or fungi. It is chemically stable over geological timescales. And it is hydrophobic, meaning it repels water, which makes the cell walls impregnated with it, both waterproof and structurally rigid. Lignin is what makes wood hard. It is what makes wood rot slowly. And it is, as we will discover, what made the first forests an accidental geological catastrophe of global proportions. But before the first forests, before even the first trees, we need to pause at one of the most remarkable and least known episodes in the entire history of life. Because in the early Devonian, before true trees existed, before true wood had been invented, there was something living on land that reached heights of 8 meters and dominated the terrestrial landscape for millions of years. And it was not a plant. It was not an animal. It was a fungus, or possibly an alga. Scientists are still not entirely certain. What they are certain of is that it existed, that it was enormous by the standards of its time, and that when they first described it in the 19th century from fossils found in Canada and other locations, they had no idea what they were looking at. Prototaxites. That is its name. A fossil organism known from deposits ranging from approximately 470 to 360 million years ago, preserved as cylindrical trunks up to 1 meter in diameter and potentially 8 meters in height, with an internal structure of interwoven tubes unlike anything in the modern world. For most of the time since its discovery, Prototaxites was classified as a conifer, a seaweed, a lichen, and a liverwort at various points, because its anatomy does not match any modern organism. The current scientific consensus, based on carbon isotope analysis that was not available to earlier researchers, is that Prototaxites was almost certainly a fungus, possibly growing in partnership with photosynthetic organisms in a massive lichen-like association. If that interpretation is correct, then the tallest organism on land during much of the early Devonian was not a plant. It was a fungus the size of a telephone pole, standing alone on a bare red landscape, the most prominent living thing on the surface of the continents for millions of years. And it has been almost entirely forgotten by everyone except specialists in Paleozoic botany. The existence of Prototaxites tells us something important about the nature of ecological colonization. Evolution does not follow a predetermined script. it does not work toward an end goal. The early Devonian land surface was an empty ecological opportunity, and the organisms that first exploited it were not the ones that would eventually dominate. The giant fungal pillars of Prototaxites gave way over millions of years to increasingly sophisticated vascular plants. Not because there was a plan, but because vascular plants, once they developed the specific set of innovations required to grow tall and reproduce effectively on land, were simply better at the game of terrestrial life than anything that had come before. Among those innovations, perhaps the most critical after vascular tissue and lignin was the root. True roots, with complex branching architecture and root hair cells that dramatically increase the surface area for water and nutrient absorption, appeared in land plants during the Devonian, and their appearance was one of the most geologically consequential biological events in the history of the planet. Roots do several things simultaneously that transform the relationship between living organisms and the rock beneath them. They anchor the plant against wind and gravity, allowing taller growth. They extend the plant's reach for water and nutrients into a three-dimensional soil volume rather than just the surface. They release organic acids that chemically dissolve mineral grains in the surrounding rock, accelerating the process of chemical weathering by orders of magnitude compared to purely abiotic weathering. And they add organic matter to the mineral substrate as they grow and die, beginning the process of soil formation that had previously been limited to the thin biological crusts of early microbial communities. The impact of root evolution on the chemistry of the Earth's surface was profound and rapid. Where early vascular plants had barely scratched the surface of terrestrial rock with their simple rhizoids, the deep root systems of Devonian plants penetrated meters into the substrate, physically breaking rock with the force of their growth and chemically dissolving it with their acid secretions. Weathering rates increased dramatically. Nutrients, particularly phosphorus and calcium, were released from rock in quantities that had never been available to terrestrial ecosystems before. And silicate minerals, which consume carbon dioxide as they weather in a reaction that produces carbonate ions and dissolved silica, were being attacked by root acids across enormous areas, driving a drawdown of atmospheric CO2 on a scale that the simple physical weathering of the pre-plant world had never achieved. This CO2 drawdown would have consequences, enormous global extinction-level consequences. But before we get to those, we need to talk about what the forests actually looked like, because the image most people carry of ancient forests is wrong in ways that are actually more interesting than the correct picture. In upstate New York, in a small town called Gilboa in the Catskill Mountains, there is a geological site that has been known to science since the 19th century, and that holds the distinction of containing the oldest fossil forest in the world. The Gilboa fossil forest, preserved in rocks dating to approximately 385 million years ago, was first discovered when quarry workers cutting stone for the construction of the New York City water supply infrastructure in the 1860s unearthed a series of large fossilized stumps still in their original rooted position, standing in the rock as they had grown nearly 400 million years ago. The stumps caused considerable scientific excitement at the time and have been a subject of paleobotanical research ever since. But for more than a century, scientists knew only the stumps. The upper portions of the trees, the trunks and branches, and whatever grew at the top were unknown because the stumps and the upper portions were preserved in different rock layers that had never been found in association. That changed in 2004 when a flood control project in Gilboa exposed new rock faces that preserved both stumps and upper portions in the same deposit. And the complete picture that emerged was startling. The Gilboa trees, which belonged to a genus called Eospermatopteris, had a structure utterly unlike any modern tree. Imagine a palm tree crossed with a fern, scaled up to 8 or 10 meters in height. The trunk was unbranched, with a swollen base, tapering upward, covered in a pattern of leaf scars that showed it had once borne a crown of frond-like appendages at the top. No branches, no twigs, no canopy in the modern sense. Just a tall, unbranched stem topped with a spreading crown of fern-like fronds. Walking through a Gilboa forest would have felt nothing like walking through any modern forest. There would have been no shade in the modern sense, just columns of vegetation casting narrow shadows at midday with open sky between them. The ground between the tree trunks would have been covered not with grass, which did not exist, but with a carpet of smaller plants, early ferns, horsetails and lycopods, in a layered understory beneath the frond crowns above. The Gilboa trees were not alone in the late Devonian forest. Alongside Eospermatopteris grew a much more significant tree, one that appears in the fossil record at roughly the same time, and that represents a genuinely different and ultimately more influential evolutionary lineage. Archaeopteris. The name means ancient fern, and it was given before scientists fully understood what Archaeopteris was, because its foliage, preserved in enormous quantities in Devonian deposits across the northern hemisphere, looked convincingly fern-like, with broad, flat leaflets arranged on branching fronds. But Archaeopteris was not a fern. It was a tree, a large tree, reaching heights of up to 30 meters in favorable conditions, with a proper branching crown, a proper woody trunk with true wood in the modern sense, a trunk that produced annual growth rings, a root system that extended deeply into the substrate, and a canopy architecture that created genuine forest shade beneath it. Archaeopteris was, in every meaningful sense, the world's first modern tree. Not first tree in the sense of first tall plant. Not first tree in the sense of first woody plant. First tree in the sense of the first organism that would be recognizable as a tree to a modern observer without specialist training. Walk into an Archaeopteris forest 370 million years ago, and you would see something that looked, from a distance, strikingly like a temperate deciduous forest. The trunks would be familiar. The branching crown would be familiar. The dappled shade on the forest floor would be familiar. The sound of wind through broad, flat leaves would be familiar. You would feel a sense of recognition, of almost familiarity that no earlier terrestrial landscape would have provided. But get closer, and the differences would accumulate. The leaves, those broad, flat leaflets that looked so familiar from a distance, were arranged in a pattern not quite like any modern leaf arrangement. The bark texture was subtly different from any modern tree bark. And if you waited through the autumn, if there was an autumn in that location, you would not see the seeds that fall from modern trees. You would see spores. Because Archaeopteris, for all its tree-like appearance and its true wood and its modern branching architecture, reproduced like a fern. It released microscopic spores that were carried by wind or water to new locations, where they germinated into tiny free-living organisms that then produced the sex cells that, when they met water, fertilized each other to produce a new Archaeopteris plant. The entire reproductive cycle depended on liquid water for fertilization, which meant that Archaeopteris, however large and impressive, was ultimately tied to moist environments for reproduction in a way that would later severely limit its ability to colonize dry ground. This reproductive limitation is worth dwelling on because it illustrates one of the most important principles in evolutionary biology: the difference between ecological success and evolutionary durability. Archaeopteris was enormously successful in its time. It dominated the late Devonian forests of the Northern Hemisphere for millions of years. Its fossils are found on every continent that was in the temperate and tropical zones during the Devonian. It was, by any measure, the dominant tree of its age. And yet, it left no direct descendants. Every tree alive today, every pine and oak and maple and redwood, belongs to a lineage that evolved after Archaeopteris went extinct at the end of the Devonian, and none of them are Archaeopteris's direct evolutionary children. The first modern tree was an evolutionary dead end. This happened because Archaeopteris's spore-based reproduction, while adequate for colonizing the moist environments where it thrived, was fundamentally vulnerable to the kind of environmental change that the Devonian was about to deliver. And the agent of that change was, with profound irony, Archaeopteris itself. Because the forests that Archaeopteris dominated were, in the process of growing, changing the planet's climate system in ways that would ultimately destroy the conditions that allowed Archaeopteris to flourish. Here is the mechanism, laid out as clearly as possible, because it is one of the most important and least appreciated causal chains in the history of life. As Archaeopteris forests spread across the Devonian continents, their root systems penetrated deeply into fresh rock, accelerating chemical weathering on a continental scale. Chemical weathering of silicate rocks consumes carbon dioxide, drawing it from the atmosphere and incorporating it into carbonate ions that eventually make their way to the ocean and are deposited as carbonate sediment on the seafloor. At the same time, the trees themselves were doing what all photosynthesizing organisms do, pulling carbon dioxide out of the atmosphere and converting it to organic matter in their tissues. A forest is, from a carbon cycle perspective, a carbon storage device. Every gram of wood in every tree trunk represents carbon that was once in the atmosphere as carbon dioxide and is now locked in biological material. And the Devonian forests were producing wood in quantities that the Earth had never seen before. The combined effect of accelerated silicate weathering and biological carbon sequestration was a dramatic decline in atmospheric carbon dioxide concentrations. The early Devonian atmosphere contained carbon dioxide levels perhaps 10 to 15 times higher than the pre-industrial modern level. By the end of the Devonian and into the early Carboniferous, carbon dioxide had fallen to levels approaching modern values, possibly even lower. This was a fall of more than 90% in atmospheric carbon dioxide over roughly 60 million years, driven largely by the spread of Devonian forests. The greenhouse effect weakened. Global temperatures fell. The warm, ice-free Devonian world began its transition toward the glaciated world of the Carboniferous and Permian. But the cooling was not the only consequence. The root weathering of rocks also released enormous quantities of nutrients, particularly phosphorus, into river systems that carried them to the ocean. In the modern world, we are familiar with the consequences of excess nutrient input to coastal waters from agricultural runoff. Algal blooms, oxygen depletion, fish kills, the formation of coastal dead zones. The Devonian forests were running a global version of this process. As they spread, their root systems released more and more phosphorus from weathered rock, and rivers carried that phosphorus to coastal seas that had never before received nutrient inputs at anything like this scale. Algal blooms proliferated. As the algae died and sank to the seafloor, their decomposition consumed dissolved oxygen, expanding anoxic zones in the deeper water. The elaborate reef systems of the Devonian seas, built by stromatoporoids and rugose corals over millions of years, found themselves in water that was warming from greenhouse changes, acidifying from carbon dioxide uptake, and losing oxygen from the nutrient-driven eutrophication that the forests were driving. The reefs collapsed. 70 to 80% of marine species went extinct. The late Devonian mass extinction, one of the 5 most severe in the history of complex life, was caused in significant part by the forests, by trees, by Archaeopteris and its contemporaries doing nothing more dramatic than growing, spreading, weathering rock, and dumping nutrients into rivers. The most destructive invasive species in the history of the Earth was not a predator. It was a tree. The survivors of the late Devonian extinction found themselves in a world that the forests had fundamentally transformed. The atmosphere had less carbon dioxide. The climate was cooling. The land surface was covered in many regions by a deep layer of something new: soil. Real biological soil, with accumulated organic matter and complex mineral chemistry, and the microbial communities that the presence of plant roots had fostered. This new soil was a resource. It was the substrate in which the next generation of land plants could grow, and it was far more hospitable to plant growth than the bare mineral substrates that the earliest land colonizers had faced. The forests had, in transforming the planet's chemistry and climate and creating a catastrophe for marine life, simultaneously created the conditions for a new and more diverse terrestrial world. The Carboniferous period, which followed the Devonian beginning approximately 359 million years ago, was the age when the terrestrial ecosystem that the Devonian forests had seeded came to full flowering, literally and figuratively. The Carboniferous world is famous for its coal deposits, those thick black seams of compressed organic matter that powered the Industrial Revolution and that continue to fuel a significant fraction of global electricity generation today. And the source of those coal deposits was forests, Carboniferous forests, which were different in character from the Devonian forests that preceded them, but were continuous with them in evolutionary lineage, growing in the warm, humid, oxygen-rich conditions of the Carboniferous tropics, producing wood in quantities so enormous that even millions of years of burial and compression have left a geological record visible to anyone who digs a mine in the right place. The Carboniferous forests were dominated not by trees in the modern sense, but by organisms that have no living relatives of comparable ecological importance. The Lycopsids, or scale trees, which reached heights of 50 meters and formed dense canopy forests in the tropical swamps of the Carboniferous. Lepidodendron and Sigillaria, the iconic scale trees, were strange and wonderful organisms. Their trunks were covered in a distinctive diamond-shaped pattern of leaf scars, created as the photosynthetic leaves fell from the growing trunk and left their imprints in the bark. They grew rapidly, reached their full height quickly, and then reproduced, producing a single massive reproductive structure at the top of the trunk, and then dying, falling, and being replaced by the next generation. They were simultaneously tree-like in scale and fundamentally unlike any modern tree in their growth strategy, more like giant monocarpic herbs than like the long-lived woody perennials we associate with the word tree. But the strangest consequence of those Carboniferous forests had nothing to do with the trees themselves. It had to do with what the trees were doing to the air. Because all that buried, unrotting wood represented something else besides stored carbon. Every carbon atom locked in an undecomposed log was a carbon atom that had been taken out of carbon dioxide during photosynthesis. And when you remove carbon from carbon dioxide without returning it through decomposition, what you leave behind is the oxygen. The oxygen that photosynthesis releases as a byproduct. And with wood accumulating across millions of square kilometers of Carboniferous swamp forest, without being oxidized back through decomposition, oxygen was building up in the atmosphere at a rate the Earth had never seen before. The numbers are extraordinary. Modern atmospheric oxygen sits at 21%. In the early Carboniferous, it was probably around 15%, slightly lower than today. By the late Carboniferous, roughly 300 million years ago, it had climbed to somewhere between 30 and 35%. Some estimates push it even higher. 1/3 of every breath of Carboniferous air was oxygen, nearly double what it is today. And that change in atmospheric chemistry had consequences that seem almost impossible until you understand the physics. Oxygen dissolves into the tissues of insects through a passive diffusion system, a network of tubes called tracheae that carry air directly from the outside world to the cells that need it. This system works fine at modern oxygen concentrations, but it has a size limit. At 21% oxygen, the diffusion gradient falls to 0 at roughly the scale of a large modern insect. Beyond a certain body size, the tracheal system simply cannot deliver enough oxygen to support active metabolism. This is one of the main reasons insects today are small. The atmosphere imposes a ceiling on their size. Double the atmospheric oxygen concentration, and that ceiling rises dramatically. At 30 to 35% oxygen, the diffusion gradients that drive oxygen transport through tracheal systems remain steep enough to supply active metabolism in insects far larger than anything alive today. And the Carboniferous fossil record preserves exactly what you would predict: insects of extraordinary size. Meganeura, a dragonfly relative with a wingspan of 70 centimeters, roughly the size of a modern crow, patrolling the Carboniferous swamps on wings that would have made an audible sound in flight. Archaeopteris, a millipede relative reaching 2.5 meters in length, the largest land arthropod that has ever existed, moving through the forest understory like a small car. Giant cockroaches, giant mayflies, a world of enormous insects made possible entirely by the excess oxygen that the unrotting forests were pumping into the atmosphere. But here is where the story turns dark. Oxygen does not just make animals larger, it makes things burn. At modern oxygen concentrations, fires require a source of ignition and reasonably dry fuel, and they spread at rates constrained by the availability of both. At 35% oxygen, almost anything will burn. Fires spread faster, burn hotter, and are almost impossible to extinguish without rain. The Carboniferous coal measures preserve abundant fossil charcoal, called fusane by geologists, far more than any other geological period. The Carboniferous world burned, regularly, extensively, catastrophically, by modern standards. Lightning strikes in a 35% oxygen atmosphere did not produce small local fires that burned themselves out against wet ground or sparse fuel. They produced continent-scale conflagrations that swept across the swamp forests, killing enormous areas of vegetation. And then, as the charred landscape recovered, fueling the next cycle of growth and burial and oxygen release. The high oxygen atmosphere also appears to have been a factor in the end Carboniferous and early Permian extinctions of many of the giant lycopside tree species. As the climate dried in the late Carboniferous and the tropical coal swamps fragmented, the combination of drying conditions and a highly oxidizing atmosphere made fire a dominant ecological force. The scale trees, adapted to permanently waterlogged swamp environments, could not survive in a landscape increasingly shaped by fire. They retreated, fragmented into isolated populations, and eventually went extinct, leaving their compressed, charred remains as the coal seams that powered the Industrial Revolution. Even their extinction was, in a sense, a gift to the future. The oxygen spike did not last. As white rot fungi gradually evolved the enzymatic capability to digest lignin during the late Carboniferous and Permian, wood began to decompose. The great organic carbon accumulation of the coal measures slowed and then stopped. Oxygen levels began to decline, reaching roughly modern values by the Triassic. The giant insects shrank to the sizes we know today. The world of enormous dragonflies and 2-meter millipedes was over, replaced by a more familiar scale of arthropod life. But for 60 million years, the simple fact that wood could not rot had produced an atmospheric chemistry experiment of planetary scale, with consequences ranging from the size of dragonfly wings to the temperature at which forests burned to the evolution of the first truly fire-adapted plant communities. And it left us, buried in the rock, all the coal we would one day need to discover fire in an entirely different sense. Alongside the lycopsids grew giant horsetails, reaching heights of 15 to 20 meters, enormous tree ferns with trunk diameters of half a meter, and the first seed plants, which were beginning to appear in the late Devonian and early Carboniferous, and which would eventually, over hundreds of millions of years, come to dominate terrestrial plant communities worldwide. The seed. The invention of the seed was, after the colonization of land itself and the invention of wood, perhaps the most significant evolutionary innovation in the history of land plants. And understanding why requires understanding the fundamental limitation of spore-based reproduction in a terrestrial environment. A spore is, in essence, a single haploid cell containing half the normal genetic complement of the parent plant, protected by a tough outer wall. When a spore lands on a suitable substrate, it germinates into a tiny organism called a gametophyte, which is the generation that produces the sex cells. In ferns and their relatives, the gametophyte is a small, flat, photosynthetic structure, usually only a few millimeters across, that is completely independent of the parent plant. It produces both eggs and sperm, and the sperm must swim through a film of liquid water to reach and fertilize the egg. Without liquid water, fertilization cannot occur. Without fertilization, no new plant can be produced. The entire reproductive cycle is hostage to the availability of liquid water on the ground surface, which means that spore-producing plants can never fully escape the humid environments near water. They can grow in drier places, but they cannot reproduce there. The seeds solved this problem elegantly. A seed is not a single cell. It is a complete embryo, already fertilized, already containing all the genetic material of both parents, wrapped in a protective coat, and provisioned with a supply of stored nutrients to fuel the early stages of germination. The fertilization step, which in fern-like plants requires liquid water for the swimming sperm, was moved inside the parent plant in seed plants, occurring within a protective structure called an ovule, where the sperm, carried by pollen rather than swimming independently, can reach the egg without requiring a film of water on the plant's surface. This was an evolutionary transformation of enormous practical significance. It meant that a seed plant could reproduce in environments where ferns could not, in seasonally dry habitats, on exposed rock faces, in continental interiors far from permanent water. The seed liberated plant reproduction from water dependence in the same way that the amniotic egg, evolving around the same time in early reptiles, liberated animal reproduction from water dependence. Both innovations enabled the full biological colonization of the terrestrial world. The first seed plants, the seed ferns or pteridosperms, appeared in the late Devonian and diversified through the Carboniferous. They looked superficially like ferns, with large divided fronds, but their reproductive structures were seeds rather than spores. The seed ferns gave rise to multiple lineages that would dominate terrestrial ecosystems for hundreds of millions of years. The cycads, which appeared in the Permian and still exist today as a small group of slow-growing tropical plants. The conifers, which appeared in the late Carboniferous and Permian and became the dominant trees of the Mesozoic before being partially displaced by flowering plants. The ginkgo, whose single surviving species is one of the most remarkable living fossils on Earth, essentially unchanged from Jurassic specimens over 150 million years old. And the nettophytes, a small and enigmatic group whose relationship to other seed plants is still debated. The conifers, in particular, deserve attention because they represent one of the most successful plant lineages in the history of land plants, and because their success is built on a specific set of adaptations that allowed them to colonize environments hostile to other plants. Conifer leaves, the needles that most people associate with the group, are an adaptation to cold and drought. Their small surface area reduces water loss. Their thick, waxy cuticle provides additional waterproofing. Their ability to remain on the tree through winter, continuing to photosynthesize on warm winter days, gives conifers a competitive advantage in cold temperate and boreal environments where the growing season is too short for deciduous trees to be efficient. The conifer body plan was good enough to persist for over 300 million years with relatively little fundamental modification, which is either a tribute to its elegance or a comment on the stability of the ecological niches it occupies, depending on your perspective. But the most dramatic chapter in the evolutionary history of land plants had not yet been written when the conifers rose to dominance. That chapter begins in the Cretaceous period, approximately 140 million years ago. with the appearance in the fossil record of the first flowering plants. The Angiosperms. The most species-rich group of land plants alive today, comprising roughly 300,000 described species, more than all other plant groups combined. The group that includes every fruit and vegetable you have ever eaten, every broadleaf tree in every temperate forest, every grass on every lawn and prairie and savanna, every flower that has ever attracted a bee or a butterfly or a hummingbird. The angiosperms did not just change the plant world. They changed the animal world. They changed the insect world. They changed the climate. And they did it so fast, in geological terms, that Charles Darwin called their origin an abominable mystery, and spent years trying to understand how such a diverse group could have appeared so suddenly in the fossil record. The flower is, at its core, a reproductive structure. It is a modified shoot in which the leaves have been transformed into petals, sepals, stamens and carpels, the structures that attract pollinators, produce pollen, and contain the ovules that will become seeds. But the flower is not merely a reproductive structure. It is an interface, a negotiating surface between the plant and the animal world, a structure designed by natural selection to attract specific animals, to load those animals with pollen, and to ensure that the pollen is delivered to another flower of the same species. The flower is, in other words, the solution to a problem that had plagued land plants since they first began reproducing sexually on land. How to ensure that pollen reaches the right destination in an environment where wind dispersal is inefficient, unreliable, and wasteful. Animal pollination, driven by flowers that reward their pollinators with nectar and pollen, is dramatically more efficient than wind pollination for many plant species, allowing them to maintain genetic diversity even when individual plants are widely separated in a landscape. The co-evolutionary relationship between flowering plants and their pollinators is one of the most intricate and beautiful examples of evolutionary interaction in the natural world. Flowers evolved shapes, colors, scents, and reward structures calibrated to the sensory systems and feeding behaviors of specific pollinator groups. Insects, birds, and bats evolved mouthparts, color vision, olfactory systems, and feeding behaviors calibrated to exploit the rewards that flowers offered. Each party in this relationship was under selection pressure from the other, and the result, over tens of millions of years, was an explosion of diversity on both sides, with new flower forms and new pollinator specializations tracking each other through evolutionary time in a process called co-evolution that has produced some of the most elaborate and improbable biological structures in the living world. The tongue of a hawk moth exactly the right length to reach the nectar in a specific orchid. The precisely shaped entrance of a fig that admits only the fig wasp that pollinates it. The ultraviolet nectar guides in a flower that are invisible to human eyes but brilliantly obvious to a bee's UV-sensitive vision. All of these are products of co-evolution between flowering plants and their animal partners, a process that began in the Cretaceous and has been running without interruption ever since. The Cretaceous diversification of flowering plants had consequences that extended far beyond the plant and pollinator communities directly involved. The spread of angiosperms across the Cretaceous landscape changed the structure of terrestrial ecosystems at every level. Flowering plant communities, with their diverse leaf forms, complex canopy architectures, and abundant fruit production, were ecologically richer and more structurally varied than the conifer-dominated forests they partly replaced. Insects diversified explosively in response to the new feeding opportunities that flowering plants provided. The diversification of insects drove the diversification of the small insectivorous vertebrates that fed on them, including the early mammals and birds that were already present in the Cretaceous, but that radiated dramatically in the Paleogene after the end Cretaceous extinction removed the dinosaurs from the ecological stage. The fruits of flowering plants were a new food resource that selected for enlarged brains and enhanced color vision in the primates that ate them. Adaptations that would later, in a lineage no Cretaceous botanist could have predicted, produce the cognitive abilities of Homo sapiens. Pull the thread far enough, and you find that the cognitive capacity that allows a human being to contemplate the history of plant evolution is itself, in a meaningful sense, a product of plant evolution. The color vision that allows us to see the red of an apple or the yellow of a flower was selected for in primate ancestors who were fruit eaters in Angiosperm forests. The enlarged neocortex that allows us to reason and plan and remember was selected for in a social primate that depended on tracking the seasonal availability of flowering plant fruits across a complex landscape. We are, more than most people realize, shaped by the plants we evolved alongside. But there is another, more immediate sense in which the history of plant evolution is the history of our own existence, and it has to do with the air we breathe. The oxygen in Earth's atmosphere, the 21% of the atmosphere that is oxygen rather than nitrogen or carbon dioxide, is a biological product. It was produced by photosynthesis, by the splitting of water molecules by photosynthetic organisms over more than 2 billion years of Earth history. And the dramatic increase in atmospheric oxygen that occurred during the Carboniferous period, when oxygen levels may have reached as high as 35% compared to our modern 21%, was driven primarily by the burial of wood. Wood that could not be decomposed because the fungi that digest lignin had not yet evolved their full ligninolytic capabilities, and so fell into swamps and was buried by sediment and was preserved as organic matter rather than being oxidized back to carbon dioxide. Every molecule of oxygen in the modern atmosphere that came from Carboniferous wood burial represents a carbon atom that was removed from the atmospheric carbon cycle and locked in coal. The coal and the oxygen are two sides of the same biological equation. This brings us to one of the strangest and most consequential facts in the entire history of land plants. The reason the Carboniferous coal measures exist, the reason we had a fossil fuel economy for the last 200 years, the reason the Industrial Revolution was possible, is that for approximately 60 million years, from the late Devonian through the Carboniferous, wood could not rot. The organisms capable of efficiently decomposing lignin, the white rot fungi, had not yet evolved the enzymatic machinery to break down the complex polymer. Wood fell, was buried, accumulated in layer upon layer, was compressed and heated over geological time, and became coal. When the white rot fungi finally evolved their ligninolytic enzymes, sometime in the late Carboniferous or Permian, the age of coal formation effectively ended. The world's coal deposits are almost entirely Carboniferous and Devonian in age, because those were the periods when wood was being produced in enormous quantities by organisms that nothing could decompose. The Industrial Revolution was powered by a 60-million-year window of biological inadequacy in fungal enzyme biochemistry. The history of land plants, from the first algal films on Silurian rock to the Angiosperm forests of the modern world, is ultimately the history of how the Earth's surface was transformed from a sterile mineral substrate into the rich, complex, biologically productive environment that we take for granted today. Every gram of soil on the planet is partly a product of plant activity. The oxygen in every breath we take is a product of plant photosynthesis. The coal and oil that powered industrial civilization are products of plant carbon fixation. The fruits and grains and vegetables that feed 8 billion people are products of Angiosperm evolution. The paper this might be printed on, the wood in every building, the cotton in your clothing, the medicines derived from plant compounds, the climate stability of the Holocene within which human civilization developed, all of these trace their origins, directly or indirectly, to the moment in the Silurian when the first vascular plant pushed its simple leafless stem above the surface of a rock at the edge of a shallow sea, and began the 400-million-year project of making the continents habitable. There is a stump in Gilboa, New York, 385 million years old, still in the ground where it grew, rooted in rock that was mud when the tree that produced it was alive. The tree itself is gone, long since decayed or compressed into organic matter that was itself eventually transformed by time. But the stump remains, a cast of the base of a trunk in the sediment that surrounded it, a shadow of wood in stone. When you see photographs of it, you do not see an alien object from an incomprehensible past. You see something recognizable, a tree, or the ghost of a tree, something that grew upward, that reached for light, that had roots and a trunk, and presumably somewhere above the preserved base, leaves of some kind, performing the same photosynthetic transaction with sunlight that every plant performs today. That recognition is not coincidental. The Gilboa trees were our predecessors in the most practical sense. They built the soil we farm. They drew down the carbon dioxide that cooled the Devonian climate and set in motion the conditions that would produce first the Carboniferous forests, and then the Permian seed plants, and then the Mesozoic conifers, and then the Cretaceous flowers, and then the Cenozoic diversity of trees within which our own evolutionary lineage developed. They changed the atmosphere. They changed the ocean chemistry. They changed the distribution of nutrients across the planet's surface. They, more than any other group of organisms in the history of multicellular life, made the terrestrial world what it is. And we are, right now, removing them at rates that have no precedent in the geological record since the Devonian itself. The world is losing roughly 10 million hectares of forest per year to deforestation and land conversion. The Amazon, the Congo, the forests of Southeast Asia are being reduced at rates that, if continued, will see significant fractions of the world's tropical forests gone within decades rather than centuries. The implications for carbon cycling, for soil stability, for the water cycle, for biodiversity, and for the climate system that supports modern human civilization are not speculative. They are grounded in exactly the science we have been discussing, the science that tells us what forests do to planetary chemistry, because the geological record has preserved the before and after of every major forest expansion and contraction in the history of the Earth. The Devonian tells us what happens when forests spread. Carbon dioxide falls, climate cools, soils deepen, nutrient runoff increases. Marine ecosystems are stressed. Life reorganizes around the new conditions. The process takes millions of years and involves extinctions and reorganizations that are catastrophic from the perspective of the organisms experiencing them, but that ultimately produce a richer, more diverse, more productive world. We are running the experiment in reverse, removing forests, releasing carbon, warming the climate, reducing soil stability, and we are running it at rates that make the Devonian transformation look glacially slow. The Devonian forests took 60 million years to change the planet's carbon dioxide concentration by 90%. We have changed it by nearly 50% in 200 years. The geological record does not give us many examples of what happens when the carbon cycle is disrupted this fast, because the Earth has rarely seen disruption at this rate. The closest analogues, the flood basalt events and the end Permian extinction, are not encouraging. But here is the thing about the history of land plants that the geological record also tells us clearly. Life is tenacious. The end Devonian extinction killed most marine species and devastated terrestrial ecosystems. The end Permian came close to sterilizing the planet entirely. The end Cretaceous killed the dominant terrestrial animals of the Mesozoic. And each time, life recovered. The forests returned. New plant communities colonized the devastated landscapes. New evolutionary radiations filled the empty ecological space. The Earth did not stay broken. It healed, given time, given the resilience of the microbial and plant communities that survived each catastrophe, given the inexhaustible creativity of natural selection operating over millions of years. The question is not whether forests will recover if we destroy the ones we have now. They will, eventually. In 10 million years, 20 million years, there will be forests on Earth as there have always been forests on Earth since the Devonian. The question is whether the civilization that depends on the forests we have now, the stable climate, the productive soil, the clean water, the biodiversity that underpins ecosystem function, whether that civilization will survive the transition. The stump in Gilboa survived 385 million years. It survived the extinction that killed the tree that grew from it. It survived the Permian catastrophe, buried and protected in rock. It survived the dinosaurs and the asteroid that killed them. It is still there in upstate New York, in the rock, a record of a tree that grew when the world was young and land was new. What survives of us will be the record we leave in the rock. And the forests we save or destroy will be a significant part of that record, written in carbon isotopes and pollen assemblages, and the presence or absence of wood in the sediments of the next million years. The first trees took 400 million years to build the world we live in. We have a few decades to decide whether we understand what that means.